Good morning and welcome. My name is Ian Lipkin. I'm the John Snow Professor of Epidemiology at Columbia University in the Center for Infection and Immunity. This broadcast is coming to you from the campus of Columbia University in the city of New York. I'd like to say it's a beautiful day here in New York, but there is some rain, but there's a little bit of blue just peeking through, which I think is a wonderful metaphor for the morning. I'd like to introduce up here at the desk my colleagues in this study. Not everyone could attend today, but we have a good representation. Harvey Alter. Raise your hand, Harvey, so everyone knows who you are. Judy Mikovits, Frank Raschetti, and Mady Hornig. Harvey is with the NIH. Judy is with Private Consulting Group, formerly with WPI. And Frank Raschetti is also with the National Institutes of Health. I'd also like to acknowledge the help we've had today from our coordinators, Amir Bhatt, Afia Genfi, and Sikan Akpan. I'm going to begin today with a brief overview of the study, and we'll follow that with a question and answer period. Now, we know that many of you who are not here would also like to pose questions. You're going to be able to do so. The journalists among you already know the website, but for those of you who are calling in from outside or writing in from outside, it's www.cii.columbia.edu. That's World Wide Web, Center for Infection Immunity, columbia.edu. Look at the latest news channel, and that will take you to our broadcast and give you an opportunity to pose questions. Chronic fatigue syndrome, myalgic encephalomyelitis, is an unexplained incapacitating syndrome that afflicts at least 1 million people in the United States. For many years, there's been the thought that it could be attributed to an infectious process of some sort, because many of the individuals with this syndrome have malaise, night sweats, lymphadenopathy, sore throat, and fever. And there have been a wide range of microbes that have been proposed as causative agents: herpes viruses, bornoviruses, Borrelia, but none has really captured the attention of the scientific community and of the patient population more than retroviruses. And in fact, recent reports relating to those has resulted in patents, diagnostic tests, therapeutic regimens, promises of vaccines, and concern over the safety of the blood supply. The two key papers relating to retroviruses were published in reports by colleagues, some of my colleagues here today. Initially, Judy Mikovits, Frank Raschetti, and their colleagues at the WPI and at the NCI in 2009, and then Harvey Alter and Shiloh and others at the National Institutes of Health a few months later. Now, the viruses they discussed were somewhat different, but they really fell into fairly similar classification, and not important really to get into the details of viral taxonomy. It's simply important to say that these were retroviruses that were related, and they were distinctive in many ways. Subsequent publications from many, but not all groups that looked into the question of whether or not these viruses could be associated with these disorders, for the most part, failed to replicate those findings. Nonetheless, none of those studies had the power that was required to, incontrovertibly refute the association between those viruses and disease. Furthermore, they did not offer the investigators who had done that initial work an opportunity to use their best methods in sorting out the challenge. So, almost 2 years ago now, with support from Francis Collins, Director of the National Institutes of Health, Anthony Fauci, Director of the National Institute for Allergy and Infectious Diseases, Harold Varmus, Director of the National Cancer Institute, Thomas Frieden, Director of the Centers for Disease Control, and Peggy Hamburg, Commissioner of the FDA, we initiated a study to look into this question in an absolutely clear-cut fashion. And many of you have already received the press release that describes our findings, so I'm just going to talk about it very briefly. Before doing so, I'd like to acknowledge the people who did the work involved in this study. We had 6 clinical investigators distributed geographical sites across the United States: Anthony Komaroff in Boston, Dan Peterson in Nevada, Nancy Klimas in Miami, Jose Montoya in Palo Alto, Sue Levine in New York, and Lucinda Bateman in Salt Lake City, Utah. They worked tirelessly, first, to define the clinical criteria that will be used in the study. We used the Fukuda criteria, the Canadian criteria. We tried to hedge the study in every which way so that we could favor an infectious hypothesis, were one to be present. Why that will be important will come up in a moment. And then the laboratories were led by, efforts were led by Shailing Low at the FDA, Harvey Alter here on the podium, Bill Switzer at the Centers for Disease Control, Judy Mikovits, Frank Raschetti, and Maureen Hansen of Cornell University, who can't be with us today. The results of the genetic tests, which were chiefly PCR, and people used whatever methods they wanted to use, found no evidence of XMRV or related viruses in either the subjects with chronic fatigue syndrome, myalgic encephalomyelitis, or the controls. There was, in fact, a finding of some antibody responses. We don't really know what that means. In approximately 6% of the controls and 6% of the experimental subjects, not experimental subjects, but subjects with disease. But again, there was no association between the presence of those antibodies and disease. And we don't really know the validity of those particular tests because we don't have defined positive control sera with which to work. Now, many in the community have written me over the past 12 hours since there have been leaks about the press release and the findings of the paper with dismay, with concern that this meant the end of CFS-ME research. Nothing could be further from the truth. Everyone here at this podium, scientists around the world, are committed to solving this problem. It is likely to be a constellation of disorders, not necessarily a single agent, be it viral, bacterial, or otherwise. The samples that were collected during the course of this project will be an extraordinary resource for addressing questions related to the causes, the treatment, the management, the pathogenesis, the basic sciences of these disorders. And these samples have been collected. They are stored in ultra-low freezers. They will be released to qualified investigators who make application through the National Institutes of Health. I am also informed that there are at least 3 active grant opportunities that will allow people to get support to study these materials. So this has been a highly leveraged study. The other thing I want to say is that it took extraordinary courage on the part of Drs. Alter, Mikovits, and Raschetti to participate in the study, to see it through to its conclusion, to actually say, we now believe, based on the data that we've obtained, that this first report may have been misleading. They, too, are committed to continuing to work on this problem and on this challenge. And I can say that in all my years of doing science, I cannot think of a single instance where an investigator came back, went through this sort of a process, and then stood up and said, I want to be counted. I don't want to admit that we made an error here, but I'm committed to moving forward and being public and forthright with the science. I'm going to read one quote from Judy Mikovits, which I think is representative of what many people say who participated in the study. I greatly appreciated the opportunity to fully participate in this unprecedented study, unprecedented because of the level of collaboration, the integrity of the investigators, and the commitments of the National Institutes of Health to provide its considerable resources to the CFS community for this important study. Although I am disappointed that we found no association of XMRV PMLV to CFS, the silver lining is that our 2009 science report resulted in global awareness of this crippling disease and has sparked new interest in CFS research. I am dedicated to continuing to work with leaders in the field of pathogen discovery in the effort to determine the etiological agent for CFS. And Judy, I think that was extraordinary, and I applaud you for that. I'm going to open the floor now for questions. I'll start with people in the audience. Please pose them to me so that I can then refer them to people. What I want to make certain is that we have an orderly process by which we get these questions answered. And Sikon, you will be providing me with questions that come in via the web. Yes? Okay. Hillary, let me give you a microphone because I want to make certain that everyone can hear you.
Oh, thank you. Hillary Johnson. Dr. Lipkin, these patients have tended to be very ill for many years, even decades, some of them, and there's been a lot of evidence for infection over the years. might there not potentially be retroviral infection in tissues other than blood, such as spleen, liver, thyroid, lymph nodes, gut lymphoid tissues, etc.? Is that out of the question, or is that something to be explored in the future?
In science and medicine, you can never say never. I mean, that's part of the nature of what we do. The question, of course, is when you put a study like this in operation, you're testing work that has previously been reported. And in the initial reports, the findings were, in fact, in blood. So where would one stop? You could survey virtually the entire body, and then you could make the argument that this was not one of those individuals. We need to do another 100 and so forth. So what we try to do in designing studies to look at the likelihood of association of a factor with a disease is to examine the first paper which came out, which described this finding, to make certain that we have the power to at least replicate that with a fair margin, and that's the approach we took. But you're correct. I cannot say that there is no person anywhere in the world who is not infected with a retrovirus in some organ that we have not sampled. But I don't see any way in which we can formally and practically test that hypothesis. Would anyone like to add anything further from the group? Harvey?
Yeah, I might, again, it is theoretically possible, but I think, look, Ian made the key point that we were trying to re-evaluate existing findings and could not confirm them, but it would be extremely unusual to have a systemic illness that may have begun in any organ not to spill into the blood or into leukocyte preparation. So, while theoretically possible, is really not too reasonable.
I want to add one point in follow-up, and that is the samples to which I alluded just a moment ago will be studied, are in fact being studied using high-throughput genetic methods in an effort to find agents as well as host biomarkers that may be indicative of disease. And in fact, if there are retroviruses that we missed using these primer sets, for example, we would hope that we would capture them using these very deep sequencing methods. In addition, there will be people who will be using immunological methods. This is not work that we would do, but others have already proposed this, that would get at questions of serology and whether or not there's previous infection and immune responses that support that hypothesis. There is an independent effort. We have some 300 samples that will be studied using this approach, 150 from cases and 150 from controls. A bit less, but that's about roughly where we are. That's under the auspices of the Hutchins Family Foundation, which has an even larger cohort. In addition to doing bloods, that group is also going to be looking at metagenomics. It's going to be looking at fecal flora to see whether or not there's some changes in the microbiome, which might also be associated with disease. Next question.
Well, I'm Deborah Woroff. Just to follow up a bit on Hillary's question and also what you just mentioned about the CFS initiative, which will do flora. I'm just wondering if there's some place where one can't get in, and you've gone to so much work compiling great cohorts, both for the CFS initiative and for this project. And with HIV, a good deal of work was done with lymph glands and so forth. And I just think it would be very tempting, since you could aspirate fairly readily, to go back and do the lymph for all these people. And in a way, I'm a little surprised, although I'm not used to the confines of scientific method, that you could resist the temptation to extract a bit of lymph as you were going along. But since that was used in HIV, I just wonder if that wouldn't be a very tempting target.
Well, doing lymph node dissections is far more invasive than phlebotomy. And once again, the objective here was to follow up on studies that were based on analysis of blood. And the findings in those studies were so robust that when we used, I think it was it Mady? Was it 3 times as many subjects as we thought we needed to confirm the results in that study? We felt that we were powering it sufficiently that we could thoroughly and adequately and rigorously test the validity of those first findings. Again, this is what I do for a living. We discover viruses. So we frequently use tissue samples and other samples and so forth. But the point of this study was to test the original findings. Going forward, should there be opportunities to examine tissues, not necessarily for XMRV or PMLV, but other agents, then we would, of course, entertain joining that sort of a search. But that's not what was proposed here. That's not what we were supported to do, and I don't think it was germane to the first part of this particular topic. Now, there is an independent effort, as I say, under the Hutchins Family Foundation to support work in called CFI and CFS research. Whether that moves into analysis of tissues or not is not something to which I have insight.
You haven't decided yet.
Well, it's not me. I don't decide.
Oh.
I'm a scientist. I'm not the director of that initiative.
Oh, it's the director who decides.
The director does that in consultation with other investigators, but that's a decision that's made by them. But again, once you start moving into tissue biopsies, it becomes a very different situation. You have much higher risk of infection, bleeding. People are less likely to want to do this. So it's going to be much more difficult. And any time you make it more difficult to get samples, you wind up with a bit of a selection bias. Who's going to volunteer for that particular arm of the study as opposed to simply a blood test or a collection of fecal material or urine or something like that?
If I could make just...
Let me just
sorry.
let Dr. Alter step in for a moment.
I think one has to resist the temptation to keep the murine retroviral hypothesis alive. What you're suggesting is to say, well, it all came out negative, but maybe it's still true by doing something else. And I think rather you have to say this study was really quite definitive and has basically excluded these agents as the cause of CFS. And yet is the impetus for moving on to more extensive, deeper studies to really find the actual cause.
And I might just, and follow up to what Harvey's just said, we don't necessarily think that there's a single cause. There may be many different viruses that act in concert. Let's say, for example, we're talking about encephalitis. There are many different viruses that can cause encephalitis, bacteria. There are chemical encephalitides. There are cancer-associated encephalitides, and they're all encephalitis. This may be a syndrome that has many factors, and what we need to do is to understand the epidemiology, the pathogenesis of this disorder, and it may well be that we're going to move into some era of personalized medicine where some people will need treatment for one agent, some autoimmune process, some toxicological process, whatever is most appropriate. So we have an open mind, but what we've done is to try to collect the materials that we needed to do this in a systematic fashion. Judy, you wanted to add something?
Yeah, exactly. I mean, we so rigorously excluded our initial findings that these agents, MLV, XMRV, is in blood. And of course, a lot from the lymph system pours into the blood. We've looked at the plasma, we've looked at the serum by deep sequencing technologies, and our original work cultured it at length. It's simply not there. But now is the time to use these valuable materials and move forward. And that's what science is all about. And that's what this opportunity was for the patient population, and why I think it was so important that it was supported at the level it was by the NIH and these investigators.
Yeah, if there had not been an XMRV and a PMLV story, I very much doubt that this syndrome would have the attention it now has. So in credit, really, to the investigators here, they put a spotlight on this. It's going to have an impact for decades to come.
Can I just make a comment?
Let me just follow up. Seacon, is there anything you'd like to bring in? Okay, I have to be fair in how I take these questions. So would you read it to me? Just one moment, please.
Yeah, so we have a question from the Post. How is this study different from the previous ones? What does it give the power to refute? How do you explain the initial positive reports? What went wrong there?
Well, there are actually several questions embedded there. So the first is, how is it different? This study is different because all the investigators who were originally engaged in the reports that either proposed these viruses or refuted the presence of these viruses, the key studies are represented in the study. So you have Frank Ricchetti, Judy Mikovits, Harvey Alter, Shailing Lowe, Bill Switzer, all participated in this study. The decisions on how to define a patient as a case, that is a subject as a case who was suitable for this project, was determined by the 6 leading clinical investigators in CFS research in the nation. The decision on how to pursue laboratory tests was made by the groups that had originally reported the findings. We did not force people to use our assays, our reagents, people allowed to do whatever they wanted. The only thing we insisted upon was that the analysis be blind, and that once a decision was made as to how to call a case, and to call it positive or negative, that could not change. So that really is very difficult to do. It required an enormous amount of effort. Just the human subject's approval alone took us over 6 months to get through all of these hoops. If we had added lymph nodes, just to give you an example, we would still be in the process of trying to plan the study. So that's an example of the study design. Secondly, the study was overpowered. What do I mean by that? We didn't start with a small number of cases. We had close to 150 cases and close to 150 controls. We designed the study so that if there was a geographical bias, so that one agent was important in one part of the country but not in another, we would not be confounded. So we had 6 different catchment areas for collecting these subjects. The controls were collected in the same catchment area and within a few weeks of the time that the subject was identified. Why is that important? Because infectious agents move through communities, and one month it may be influenza that's moving through, the next month it may be strep throat. And you can be confused as to whether or not something is significant or not different. So we made certain that everything was close together, geographically, temporally, so we would have a very clean result that would be impossible to misinterpret. And we carried it through to completion and made certain that at the end of the day, everyone was in accord. In contrast with some of the earlier studies where people had they were unhappy with the way the results were presented, they didn't actually stand up and say, I'm really behind the work that was done here. This group has said, we are firmly behind these findings. Again, this is an extraordinary thing. Perhaps as important as what you're seeing with reflection, with respect to chronic fatigue syndrome and myalgic encephalomyelitis, is an example of science at its finest. It doesn't get any better than this. You put ideas up, you try to knock them down, and you move forward, and that's what everyone is saying here. So that's the way in which the study is different. Now, as to what went wrong the first time around, all I can do is say, as Judy has said, and as Frank has said, and as Harvey has said, there was an error. There was a contamination. This happens. This is not a perfect world. We do the best we can. And science is self-correcting. The important thing is that you do correct and you do move on. And again, as Judy said, the silver lining is that there's attention now on this syndrome. There will be progress made. There are supplies, reagents, techniques. There are people who are working together who've never worked together before, so we will make progress. Our first foray, if you want to call it that, into chronic fatigue syndrome was back in the middle 1990s when there was a Japanese group that said 50% of chronic fatigue syndrome in Japan was due to Borna disease virus, a virus that we'd identified several years earlier. And when we went through this and were unable to find any link between Borna virus and this syndrome, the one thing that did impress me was that there was an enormous amount of immunoreactivity that appeared to be nonspecific in these individuals. So at a time then people were saying this was a psychosomatic disorder, I said 2/3 to 3/4 of the individuals whom we've studied have polyclonal B-cell activation. They're sick. We don't know why, but they're sick. So that's really the take-home point. I mean, I think people are now aware of this. There's going to be a request for these sorts of materials. 2 people have already received samples and are beginning to work them up. So I'm optimistic that we will make headway. And this is going to be a very open application process, and we hope that people all over the world will be interested in using these samples and shedding light on this important syndrome. Now, maybe some of my panelists would like to add something. Frank, would you like to add anything? You've been quite quiet, which is unusual for you.
Well, Ian, you're so eloquent, I don't know if I can add anything. I think the important thing to say about the original studies and what's come forward is science is a self-correcting process, as Ian said, and it demands reproducibility. Where the original findings were in error is almost difficult, if not impossible, to tell at the moment, because that's a historic fact. We can't go back to those samples, those reagents, and whatnot. But the key thing is any finding needs reproducibility, and it failed on that, so we have to move forward.
And I should just say that Frank's lab and Maureen Hansen's lab went to extraordinary efforts. They cultured for several weeks, separated by space and time and location, more than 360 samples. Dan Bertolet, Carrie Petro-Sadowski, Maureen's Ying Huang, extraordinary amount of work. They cleaned, they put new filters in every hood, changed out labs. You just cannot say how much effort went into this study, and we are 100% confident in the results.
It was an enormous amount of work that was invested here. I'd like to just turn to Mady Horning for a moment. She's the Director of Translation Research at our center. She can perhaps talk about some of the studies that people are anticipating doing with these types of samples.
There are clearly a host of questions that have that remain, that we're dedicated to be pursuing, along with other investigators. We know, as Ian alluded to, there appears to be, at least in a portion of patients with chronic fatigue syndrome, and immunoreactivity, differential immune profiles. We don't know what those mean. We don't know why they exist. We don't know which subsets of patients may be most likely to have those sorts of indices. But with these sorts of study designs, we've done such exquisite characterization of patients. We've controlled for so many factors that we know could alter the infectious agents in the catchment areas, and age and sex and all of those other factors, so that we can begin to pursue what the indices might be that may lead us to some of the etiologic factors. And additionally, there will be studies of epigenetics, looking at gene expression and different RNA species that may regulate the expression of genes and provide further light on the pathogenesis of this syndrome.
Hillary? Just one second. Just getting you a microphone.
The origination of the discovery of XMRV was by, as you know, Joe DeRisi and Don Ganem at UCFS. And also there were a number of papers published in 2008, 2009 on relating XMRV to prostate cancer as well, or associating it, not saying it was the cause, obviously, but nor did your paper, Mikovits and Richetti's paper, say that it was the cause either, by the way. But I guess my question basically is, why so much emphasis, so much urgency to retract the connection between XMRV and chronic fatigue syndrome? And this, there haven't been any calls for the retraction of the prostate cancer papers that were where XMRV and prostate cancer were associated. Just curious.
So I think it's an interesting question. I don't see any delicate way for us to address it, and it's somewhat off point. So I'll be happy to talk with you about that offline, but not online. Other questions? Please.
Yes, this is Deborah Woroff again. I just, I'm very impressed, and I understand the points that you've made about the parameters of what's been done and what will be done. As someone who's had this disease for 23 years, if I look at the IRS tables for life expectancy and subtract from that the 20 years that are supposed to be sacrificed to this disease, I discover that I'm going to be dead in 45 days. Now, with that in mind, I wonder, and we all know that this disease and its investigation has taken a lot of bad turns along the way and has been subject in various departments, actually not involving any of you, but in other departments, to a great deal of negligence that has held it back. What do you think are the optimum things that can be done to really speed along the progress towards a cure within my lifetime?
So I've been involved with a number of organizations that have been successful in promoting research. The most successful, of course, was the HIV/AIDS community. And thereafter, the autism community. And, there are others that have been more successful, less successful. There's no question but that your political leadership will respond to pressure. So this may not be a very popular thing to say with everyone, and because at the National Institutes of Health we like to focus on basic science and applied science without talking about specific diseases. But the fact remains that if you say there's going to be a certain amount of work that's going to be focused in a particular area and you can focus it in that way, then people will work in that area. So I would encourage you to try to motivate your colleagues, people with this disease, related diseases, and their families and loved ones, to request additional support in these areas. This is what happened with autism, and it made a huge difference in the investment at the level of the National Institutes of Health and even in the Department of Defense. Again, this is very difficult for people who have a chronic illness, which is debilitating. It's very difficult to be aggressive in pursuing that sort of support, but I think that's really what you can do. What I don't want to see happen, and this is why I'm somewhat reluctant when we start talking about examining organs and such, is to have something where people want to donate themselves, their bodies and such. I would strongly advise against it, and I can personally tell you right now that I won't touch it. Okay, so there are examples where people have done things like that in science, and I won't encourage it, I won't condone it, and I won't analyze it. So we will only analyze samples from people who are going through some sort of a clinical trial where everybody has been consented. We're not doing any harm. Okay, this is absolutely key. Harvey, did you want to add something?
Yeah, I think what's going to accelerate things at this point is that there's been a shift in thinking. To date, it's been looking at organisms, mostly viruses, as causes of this disease. And everyone has sort of petered out with further investigation. But now, I think we're thinking of the uniqueness of the host. And why do certain people respond to common viruses in a different way? Why is there a hyper response to probably cytokine production, other things released in certain patients in response to viruses that most people handle differently? And I think now with the technology of it, you can look at all these things simultaneously, as Ian says. You can look at the entire genome for viral and infectious agents, but you can also look at host response genes and cytokine productions and everything's imaginable now. All the ohms can be looked at simultaneously. So I think the answer is going to come out of that approach, and it'll happen more rapidly than it has happened to date.
I think it will, but as someone who's not an employee of the federal government, I can tell you right now that if you make noise, people will have to respond. The National Institutes of Health, the whole scientific enterprise in this country is frankly in jeopardy because we may be moving into sequestration, and this is going to have a huge impact on the
Sequestration?
there's what happens if we don't come up with a plan for the budget.
Oh,
The projected reductions in the budget for the National Institutes of Health are going to be staggering. So people do have to have the resources to do the work. So this is a time, in fact, when you can be very aggressive and vigorous about pushing back and saying, we need good science, we need good medicine in this country, we need to push forward in that fashion. And although my colleagues who are federal employees can't say that, they will agree with me in private.
I would like to.
Oh, sorry.
I would like to say something about that. Ian and Harvey and Mady were talking about the response of the host and the immunoreactivity they find.
Yeah.
In the it's a propitious time for you to be proactive because 90% of what's known about the immune system was discovered in our lifetimes. When I got into the field, and I've tried to publish a paper on a cytokine from a T cell, all the reviewers came back and said, you're crazy. T cells don't make cytokines. So you see what's happened in the last 35 years? It's a good time to be proactive because what we know about the innate immune system, which is the orchestrator of the immune response, is much more known now than it was known 30 years ago. So the scientists need to use the new technologies like Harvey said, but the organization can be proactive because there's much more to be learned now.
More than was known 5 years ago, not 30
Yeah,
years
that's right,
ago, 4
absolutely.
years ago.
And now we have these samples to go back and
Yeah,
look
right.
as we discover new things.
And as the next-gen sequencing
Correct.
techniques
That's
are
right.
allowing us to do deeper and deeper sequencing, integrated with these other techniques.
just from Harvey's remark, I didn't quite understand whether you meant that there was already work going on in the sequencing, or not sequencing, in this area of cytokines and so forth, or you already know of someone who's planning it? I just didn't understand the timeline.
Yes, I think
Yeah,
our
Yeah.
senior member here
yeah,
is already progressing in this direction, and others will too. It's just such a logical way to go now, and the technology is what allows it to happen. So then it does get down to money, and these things are very expensive. So where is that money going to come from?
And if I may add, last week, a patient organization brought it to my attention that last week the FDA took the lead in this regard at the direction of Mr. Obama, President Obama, and said that this is a serious and life-threatening disease, and encouraging drug development in that category, bringing industry and into the field, biotechnology, big pharma, and giving them the opportunity to begin to look for drug targets in this realm and produce new therapeutics and new clinical trials.
Yeah.
Hillary?
Thank you. Quick question related to that. Dr. Lipkin, and this might be something that Dr. Mikovits might be eager to speak about, which is that this research has obviously opened up a lot of hints and clues for future avenues of therapies. The cytokine footprint or the disease footprint may have opened up new avenues with peptide T, GC-MAF, et cetera. Can you or someone on the panel talk about where therapies might be directed in the future?
So that's a very good point. So this actually harkens back to something that Dr. Alter was talking about just a few minutes ago. And I think, and Frank and Judy resonated with this as well. There are, not everyone who's exposed to the same infectious agent has the same response. This has been shown in humans, it's been shown in animal models, so we know it's a real phenomenon. And how you respond is a function of many things. Your genetic background, the way your genes have been modified, your nutritional status, what infectious agents you've seen, what toxins you've seen, and so on. You can use some of these new methods for analyzing the proteins that are present in the blood. Not just cytokines and chemokines, but other proteins that have yet to be defined. You can look at different gene products like different types of RNAs. All of these will provide insights into pathways that are differentially regulated in some people than others. And therefore, even without necessarily knowing the infectious agent, you may be able to modify the response. The response may be, in fact, what is the problem? It may not even be so much the agent per se. And the agent need not even be present at the time that the disease is manifest. So you can show with animal models, and very sophisticated animal models, that a process can be set in motion early in life that manifests decades later as cancer, mental illness, abnormal responses to other infectious agents, what have you. So the big focus really will be on integration of three components. What Mady Hornig likes to talk about is the three strikes hypothesis. The genes, the environment, and the timing, all interacting in concert to result in some outcome. And you can intervene at various steps along that process to modulate it, to improve the outcomes. Now, if people respond well to some drug, the question is why. If you can exclude a placebo effect, which can in fact be substantial, and that's why we need clinical trials, then that may provide some insights into which pathways are aberrant, and that may give you insights into how the disease process evolves and ways to prevent either disease in the first place or to mitigate it. Is that clear?
Yes.
Okay, thank you. I think we'll probably just take two more questions and that's it, unless there was something else you wanted to put forward, Sikon. Please, I can't read your name tag.
My name is Jay Spiro. Could you speak to any clinician input that you might have received in designing the study or planning it? Yes.
So the study was conceived in stages. We gathered first the laboratory people, represented here, as well as Bill Switzer and a couple of others, and we went over the various lab tests, and we identified amongst ourselves who we thought the leading clinicians might be who would be able to participate in a study of this sort. So I mentioned the names of those individuals, and there were 6 of them. Tony Komarov, who's been studying chronic fatigue now for decades, Nancy Klimas, so he's at Harvard, Nancy Klimas in Miami, Cindy Bateman in Salt Lake, Dan Peterson, who was part of the original team that described the syndrome in Incline Village, Jose Montoya, very well known to many of you, and for his work primarily on herpes viruses, and then Sue Levine, who's here in New York City. So they all met and had input not only from themselves, but also from Suzanne Vernon from CFIDS, who participated in helping to generate some ideas about what sorts of things might be important. We then came up with what we thought was the minimal definition that we would use, and then we went further. Again, what we tried to do is to make certain that the people we identified to participate in the study would be the ones who would be most likely to have an infectious agent at the foundation of the onset of the illness. So we emphasized fever, malaise, night sweats, lymphadenopathy, weight loss, and so on. All those individuals were brought in. We had laboratory tests performed. The reason we don't have 150 people in each of the two groups is that some of the individuals, despite having a history that was not remarkable for things like hypothyroidism or cryptic hepatitis or anything like that, in fact did have those disorders. So we had to delete those from the pool because we wanted to be certain that there wasn't something else that might somehow detract from the power of the study that we were doing. So the decision on who to recruit was made by the clinicians. The decision on the laboratory tests to do was made by the laboratory investigators. The decision about where to eat dinner and how to write up the paper was made by me.
And where did you eat dinner?
McDonald's. Hillary?
I think it's a question in the back.
Is there another question in the back? One more question.
There's a question from Martin Ensrink from Science.
Yes.
And the question is directed towards Mikovits and Rossetti. Can you explain why the many failed attempts to replicate your 2009 study, the Coffin Pathic paper about XMRV's origins, and the blood working group study published in 2011 failed to convince you that your 2009 paper was wrong? And what sets this new study apart?
I think we've already been over that ad nauseum.
Okay.
So we've talked about the fact that the first study was not appropriately powered. We've talked about the fact that the other studies did not allow them to do exactly what they wanted to do and the way they wanted to do it. I don't know that there's really anything else to add to that, is there?
No.
Frank or Judy?
No.
Okay, well, I think, as I say, I think we've addressed that question. Last question, Hillary.
Thank you. I'll keep this short, but I'm trying to focus on what were the good things that came from this work, and we've seen that XMRV is a chimera, as it's been described, and isn't this somewhat worrisome in that, we've seen how rapidly XMRV can replicate in human cells. We saw what XMRV did in the rhesus macaques. And we are still using MLV and gamma retrovirus research in creating recombinants and vaccine therapy. Has this experience caused people at NIH, maybe you could speak to this, or Dr. Rossetti, has this caused people at NIH to be somewhat concerned and even alarmed about the fact that these recombinants can occur in the lab, that there can be man-made chimera/viruses?
So there's a term that's used to describe this. It's called gain-of-function research. And it's come up most recently in the context of H5N1, the work that was done in Rotterdam and in Wisconsin and in Japan in generating recombinant influenza viruses that appear to have more virulence, more transmissibility in models, in ferret models. And this is an issue that's being addressed by the scientific community. There is no clear pathway for this as yet. It's being developed at very high levels, in fact, within the World Health Organization. So this is something, because whenever you're creating new viruses that have potential to infect humans, there is the issue of how that should be addressed and how that should be contained. Now, I can tell you that with respect to H5N1, in the same journal in which our work has appeared, the journal MBio, there will be a series of reports at the end of this month that tackle this issue. And there will be people speaking for H5N1 recombinant research at various levels, people speaking vociferously against it, and people who are in the middle who are talking about doing work of that sort under high-level containment. And all those different perspectives will be addressed, debated in MBio, and I encourage you to wait for that. It'll be coming up in about a week's time. And there will be a press release, and I'll probably be participating in that one as well, but it won't be here. But in any event, that is with a disease that, you know, is acute, right? It's something that causes disease very, very rapidly. But I think the same arguments, the same principles are at play, whether you're talking about something that causes acute or chronic illness. And this is something that the scientific community and the community at large is going to have to wrestle with and make decisions about. Very good point. Well, let me first thank you all for attending, and I particularly want to acknowledge my colleagues, Harvey Alter, Judy Mikovits, Frank Raschetti, Mady Hornig, and people who could not be with us, Shailing Low at the FDA, and Bill Switzer and his colleagues at the CDC, and of course all the clinicians who I've just named just a moment ago. Thank you very much.